I just, okay, got it. I'll just put the laser pointer so you can also see the laser pointer. Okay, so our team here in Paris is focusing on the study of neuronal interactions between the cerebellum and other brain structures in behavior. And today I will talk about the coupling of the cerebellum with the forebrain motor circuits and its relevance for motor disorders, and notably motor disorders such as Parkinson's disease, dyskinesia, and dystonia. So the circuits that I'll discuss today involves the cerebellum, basal ganglia, and cortex, which are the three main players of the forebrain motor circuits. So they do not work independently one from another. They have reciprocal connection. So here we can see the cerebellocortical reciprocal connection with the cortex, layer 5B neurons more precisely, that are projecting to the pontine nucleus. And then there are projections from the pontine nucleus to the cerebellar cortex and also to the cerebellar nuclei. So this is the descending pathway. And we also know that for the ascending pathway, the cerebellar nuclei are projecting to the motor thalamus, that is projecting to the cortex. So we also know that there are basal ganglia cortical loops. And in this case, the basal ganglia is projecting to another part of the motor thalamus that projects to the cortex. And more recently, it has been shown that there are cerebello-basal ganglia loops. And in this case, the cerebellum projects to another part of the thalamus, which is called intralaminar thalamus, such as centrolateral or parafascicular in rodents, and this thalamus will project to the basal ganglia. So we see that we have this heavily interconnected network. And we also know that all these structures are used by the brain in order to complete efficiently motor actions. And each of them has the ability to learn in order to optimize the success of a movement. Yet, dysfunctions in each structure yields different neurological disorders. So, for example, dysfunctions in the cerebellum yields ataxia, while dysfunctions in the basal ganglia yields Parkinson's disease, which can affect the other structures. So we are interested to see what goes wrong into the cerebellum when something goes wrong in the basal ganglia. And our hypothesis is that plasticity in the cerebellum continuously adapts the motor function, including when the basal ganglia are dysfunctional. And our objectives are to identify the plastic events involving the cerebellum in pathologies associated with the basal ganglia. And so far, we looked at the case of the Parkinson's disease, the dyskinesia, and the dystonia. And we did not only describe these plastic events, but also we used plasticities to correct the problems. And for this, we induced plasticity in the system through stimulation to treat pathologies. So we explored the benefit of cerebellar stimulation to reverse motor dysfunction. So we already know that something is happening in the cerebellum in Parkinson's disease. And in the normal state, we know that the cerebellum excites the cortex through the motor thalamus, while the basal ganglia inhibits the cortex through the motor thalamus. So a pathology developing in the basal ganglia can also show abnormal activities in the cerebellum. And we took the case, for example, of Parkinson's disease. And we know that in Parkinson's disease, we have the dopaminergic degeneration that takes place in the basal ganglia. And we know that we have a disruption of basal ganglia. So here it's a resting state image, and we can see here in red an increased activity in the pallidum, in the basal ganglia of a Parkinsonian person. And we also know that we have a disruption in the cortical function. And here we can also see here in the posterior parietal a decreased activity in blue in this resting state in Parkinson's disease. However, we see that we have increased metabolic activity here in red in the cerebellum, and this activity is consistently observed. So this is a resting state study, but in the last few years, more and more studies during movement showed cerebellar changes in the cerebellar activities in Parkinsonian patients. So I'll introduce you to some of these studies, especially imaging studies. So in this one, for example, we can see during movement in red that we have more activation in the case of Parkinson's disease in the cerebellum versus a healthy control during an automatic execution of a learned motor sequence. So here in this one, it's even more interesting since we see two movements. So we see here one moment of the movement, and here another moment of the movement. And here we can see that the activation of the basal ganglia here, and we see also in the normal, but we see also that in the Parkinsonian patient, this activation disappears, but there is evidence for an overactivation of the cerebellum compared to the normal subject at the same time of a movement, as if it compensates for the lack of the activation of the basal ganglia. So we see that this is transient, since we don't see it a little more later in this moment of the movement. And it must be linked to a movement and limited in time for that type of movement. So we see that we have this increased cerebellar activity in Parkinsonian patients that may reflect redistribution of cerebral computations. So we can also see changes in cerebellar activity, not during just a movement, but also during learning. So here, the authors perform a task, good trial and error sequence learning. So here is the learning, the number of current movements to the target. And we can see the controls and we can see the Parkinsonian patients. So at the beginning, we see that there is a difference between the two. And then they looked at the cerebral activities when the performance to achieve was equal between the Parkinsonian patients and the controls. So here at the end. And we can see here that in the case of this learned movement, we have an overactivation here in yellow of the bilateral cerebellum. And this is also coupled with overactivation in other region in the hemisphere that is ipsilateral to the movement. So we have these compensatory suractivations in the Parkinsonian patients that help the patients to perform the task as well as the controls. So here we can see abnormal metabolic activity. And here, so I told you about the cerebellum, but when we look at abnormal metabolic activity, we see in Parkinson in humans, we see that it's not only in the cerebellum, but it's in several structures. So it's in a circuit, in the cerebellum, in the thalamus, in the sensory motor cortex. And we know that in Parkinsonian disease, there is the dopaminergic degeneration in the basal ganglia. And it was not clear to what extent this is sufficient to induce brain activities that are characteristic to Parkinson's disease. And then we have this first image in a monkey, so in an animal model. And we see that a dopaminergic lesion in animals, the MPTP monkeys, can induce similar metabolic anomalies. So we see here that we have this cerebello-thalamocortical covariance that is suggestive of a functional network. So Timmerman looked even at the dynamic of the network, and here he recorded from the cerebello-thalamo-premotor motor cortical areas, and he saw that there is a dynamic in this network in patients that have a tremor in Parkinson's disease. And he showed synchronous oscillations at around 8 Hz in this circuit, so a tremor-related abnormal coupling in the cerebello-thalamocortical circuit. We also know that this pathway can be a site for therapeutical stimulations. And in this study by Gross, they recorded the activity of the cerebellocortical thalamic relay. So this is the VIM. And so the ventralis intermediate nucleus in the thalamus. And we know that this is the recipient of the cerebellum, and then he projects to the cortex. And so they record the activity of cells here, and they found cells that are particularly related to the tremor. and the stimulation had an effect on these tremors. So we see that this is a therapeutical target, this network, and targeting the cerebellothalamocortical pathway with DBS can alleviate Parkinsonian tremor. So we saw that there are abnormal activities in different structures, but how the cerebellocortical circuit is normal or abnormal. And to study the cerebellocortical network in humans, we can use transcranial magnetic stimulation. So how does it work? We can stimulate the cortex, the motor cortex, and then we look at the EMG response in a muscle, and then we can record this EMG response, so the motor evoked potential. And then we can stimulate the cerebellum and then the cortex. And we also record this motor evoked potential. And in this case, we'll see that there is a decrease. So this is called cerebellar inhibition. So this is cerebellar inhibition, and this cerebellar inhibition depends, so this decrease depends on the delay between the two stimulations. So the cerebellum has to be stimulated before the cortex to obtain the CBI. And this decrease has been interpreted as a change in excitability of the motor cortex after the stimulation. So it is due to the fact that activation of Purkinje cells, that are inhibitory cells, reduces the excitatory cerebellocortical drive. Now, what happens in Parkinson's disease? So people look in Parkinson's disease, and here you can have the control subject. So we can see this cortical excitability reduction, the cerebellar inhibition, CBI. But then if we look in Parkinsonian patients here without medication, we see that we have this decrease in the CBI, and the addition of levodopa here, the Parkinsonian patients with medication, does not restore the deficit. So the cerebellar CBI is disrupted in Parkinsonian patients, suggesting a functional impairment of the cerebellocortical pathway. So now I showed you that there are anomalous activation and activities in the cerebellocortical network in Parkinson's disease, and evidence for cerebellocortical functional disconnection in Parkinson's disease. But what is the nature of the alteration of neural activity and the functional connectivity of the cerebellothalamo-cortical loop? So to study this, we decided to do the studies, to study this phenomena in animal models where we can be more invasive. And we perform an experimental Parkinson's disease model, a classical model of 6-hydroxydopamine. So for this, we inject a dopaminergic toxin. So this is the 6-hydroxydopamine in the medial forebrain bundle that links the substantia nigra to the striatum. And then we can measure the impact of this dopaminergic lesion classically by using immunostaining of the tyrosine hydroxylase, which is an enzyme essential for dopaminergic synthesis, and it is found in dopaminergic terminals. And here we can see this diminution of the tyrosine hydroxylase here in the striatum unilaterally, since we only inject on one side. And this diminution here has an impact on motor activity. And for this, we looked at the asymmetry in the use of forepaw in the cylinder test. So this is a very easy test. We look how a mouse, or put a mouse in the cylinder, and we look how a mouse will touch the walls of the cylinder with the forelimbs. And we can see here in red, in the case of the lesioned animals, that we have more use of the ipsilateral forelimb in these lesioned animals because the contralateral is impaired because of the lesion. So we can see that we have this motor impairment of the forelimb, but we also see effects on the locomotion. So for the locomotion, we look in the open field. So we put the mouse in an open field, and then we look at his activities. And here we can see trajectories here in black from normal animals, so they go around, and in red for the lesioned animals. And we can see here the total distance traveled over weeks, in red the lesioned animals, and we see that is much reduced over weeks. So what we also looked at is at the gait test, and here we look at the same animal, before the lesion and then after the lesion with a toxin. And we could see here in red and in blue the forelimbs and the hind limbs. And we see here that after the lesion, they hesitate initiating movements. And this is interpreted as akinesia. And also they have smaller steps, and this is called fascination. So all this is consistent with the literature and confirms that we have a classical model of Parkinson's disease. So now, what is the nature of the changes in the cerebellocortical pathways in this animal model of Parkinson's disease? So to answer this question, we recorded with electrophysiology the activity in the cortex and then in the cerebellum in this animal model of Parkinson's disease. So here you see recordings from the cerebellar nuclei and for the cortex. So here is the motor cortex and here the cerebellar nuclei. So this is in freely moving mice in the case where we did the controls and also in the case where we use this toxin to have the animal model of Parkinson's disease. So these are examples. So here every time when you see a line with when the cells fires, we have a sparse firing rate in the motor cortex, and we have a higher firing rate in the cerebellar nuclei. And when we look at the data, what can we see here? So that over weeks in the awake, freely moving mice, we see the classical decrease in the motor cortex activity in the case of the lesion, but we saw an increased activity in the interpost nucleus, which is one of the nuclei that is the output of the cerebellum, and that projects more to the motor cortex. So we have a depressed motor cortex activity, but increase in interpost cerebellar nucleus. And with these results, we could ask why we have this increase tonus in the cerebellar nucleus that cannot be translated to the cortex. And we know that the activity in the cerebellar nuclei can be influenced by the cortex through the pontine nucleus, that is glutamatergic projections directly to the cerebellar nucleus, but can also be influenced by the Purkinje cells in the cerebellum that are inhibitory cells. So we asked how is the activity of the cerebellar Purkinje cell in this model. And then for this, we took advantage of a line that we developed with colleagues here in France, the L7 channelrhodopsin transgenic mice. So these mice are expressing channelrhodopsin in all Purkinje cells, so allow us to specifically stimulate sets of Purkinje cells. And here we can see that every time in blue here, when we stimulate Purkinje cells, we have an action potential. So then we do the opto-identified Purkinje cells in our animal model with the toxin injected in the basal ganglia. And for this, we did this with Fabien, who was a postdoc in the lab, and we use an optrod. And then here we can see a Purkinje cell that is normal and it fires a lot. And we can also see that during this blue period, so the blue period, because it's an optrod, it's at the moment when Fabien will excite the cell, it fires even more. And so this is the normal high firing rate. And then we have also some examples. So here in the model of Parkinson's disease, the 6-hydroxydopaminergic model, so we see that some cells fires the same, but we see that some have a low and irregular Purkinje cell firing rate, like we can see here, low irregular, and even silent. So this silent one, it's interesting because we could have them because Fabien was finding them using intermittent light pulses while moving forward with his optrod. And these cells, we can only see the complex spikes here, and he could record them because they were answering to the optogenetic stimulation. So we have this decrease in Purkinje cell inhibitory action on the cerebellar nuclei in the Parkinson's disease. So now, how is the functional state of the cerebellocortical pathway in this model? So we saw here that we can have a decrease firing in the Purkinje cell. So because we have this model, we were thinking that we'll stimulate directly optogenetically the Purkinje cell and then look to see how is the functional state of the cerebellocortical pathway. So this is something that will parallel what is done with CBI in humans. And then we know already, because we showed in another story in the team, the effect of Purkinje cell stimulation on the cerebellocortical pathway. So in this case, we could really see that if we stimulate Purkinje cells, we'll have the excitatory action on the Purkinje cells. But then when we go at the first synapse in the cerebellar nuclei, here the dentate, we can see here that the synchrony in the cerebellar cortex allows a tight control of the nuclear discharge, because we see here the inhibition while we are stimulating the Purkinje cells that are inhibitory. And then we see that the cells restart firing. And if we go at the next synapse in the motor thalamus here, the ventrolateral thalamus in mice, we can also see that some thalamic nuclei require cerebellum in order to fire. And then we went in the motor cortex, so in the layer 5 of the motor cortex, where the motor thalamus is projecting. And here also we can see excitatory response after the inhibition evoked by the cerebellar Purkinje cell inhibition. And we also checked to see if we can see this effect in the local field potential. So here we don't see much during the stimulation, but here you can see this wave that corresponds, so that is developing after, and it corresponds to a sink in the layer 5. So here in the local, we can look at the current sense density, and this says that this wave is local. So this means that with the stimulation of the Purkinje cells, we have this inhibitory excitatory sequence in the ascending pathway that is triggered by Purkinje cell excitation, and that can also be recorded in the LFP in the motor cortex. So knowing that, it's what we perform in our animal model of Parkinson's disease. So here you can see that with this good moment to explore the state, we have here examples, so of the motor cortex here, the motor cortex local field potential while we are stimulating. So during stimulation, we don't see much as before, but then after we see this wave developing, as I showed you before. And if we look at all the animals, we see that we have this depression in the cerebellocortical pathway, which is something that is consistent with the CBI that was observed in patients. So overall, in this story, we show that we have a decrease firing rate in the Purkinje cells in the case of our animal model of Parkinson's disease, but a fast firing in the cerebellar nuclei. But this was not enough to potentiate the activity in the motor cortex, where we can see this decreased activity over weeks. And also we see this reduced cortical response, which is evoked by cerebellar stimulation. So we saw that we have a presence of compensatory adaptation in the cerebellum following basal ganglia lesion, but the benefit potentially limited by the depression of the cerebellocortical pathway. So the cerebellar activity and its coupling to the forebrain are modified by pathological alterations in the basal ganglia. Now, we ask if we can use these cerebellar perturbations and their impact on the forebrain to improve the condition of patients. And we looked at levodopa-induced dyskinesia in Parkinsonian patients. So two teams showed in humans that magnetic stimulation of the cerebellum in Parkinsonian patients with levodopa therapy could significantly reduce dyskinesia. And so this is one of the side effects that we have of levodopa in many patients. And here you can see that, for example, here in black, when they administer the levodopa, we see the increase in the dyskinesia score. So these are abnormal involuntary movements. And then here in white, you can see that they only perform a protocol with theta burst stimulation less than 2 minutes a day, so 2 times 40 seconds per day during 2 weeks. And with this, they could have this decrease, that it's still a small decrease, but a decrease in the abnormal involuntary movements. So there is a functional impact on cerebellar stimulations on the levodopa-induced dyskinesia. And we get inspired by these studies because we think that in animals we can be more invasive and we can really test if we can see this and if we can propose something that can work better than for patients. And what we did with Berenice during her PhD. So we asked if we can reproduce these therapeutic rodents effects in rodents. And for this, we looked at one side effect of the levodopa, specifically the oral lingual dyskinesia that we can see here in humans, and it's something that we can also see in mice. So the protocol of Berenice was first to induce the lesion, so then we can have this Parkinsonian model for 3 weeks, and then she gave levodopa, so then we can have a dyskinetic mouse. So we'll have dyskinetic mice, and then we perform the same stimulation as the ones that done in humans, but so 2 weeks cerebellum at theta rims, but in this case, we use optogenetic stimulation of Purkinje cells specifically, because we don't know what is the target of the transmagnetic stimulations. And also we have the specificity of the orolingual zone in the cerebellum, which is the Cruz tube area, because we are looking at the orolingual dyskinesias. And we did this after we had the dyskinetic movements, and we call this, so this will be in green, we call this the corrective cerebellar stimulations. And we also added a new group where we tried to give the stimulations on the cerebellum in the same time with the levodopa, with the beginning of the levodopa, with the idea that we can prevent this, and then we call this the preventive cerebellar stimulations. So now we can look at the behavior. So these are the dyskinesia, the orolingual dyskinesia. And we can see here, so this is levodopa. And we can see here that levodopa does not induce dyskinesia, so this is normal. But we can see that in the lesion mice, the one that are the animal model of Parkinson, we have dyskinesia, that it's stable over weeks. And then we apply this protocol of optogenetic stimulation specifically on Purkinje cell orolingual zone. And we have a decrease almost down to 0 to this abnormal involuntary movement. And we can see that we can also have the effect even after the end of the stimulation. And even better, if we start the stimulation of the cerebellum in the same time with levodopa, what we could do in mice, we see that we can even prevent the apparition of such abnormal involuntary movements. So we could see this beneficial effect of cerebellar stimulation on levodopa-induced dyskinesia, and yet stronger than in patient, because we were specifically on Purkinje cells and specifically on the Cruz tube, which is the orolingual zone. So now, what is the effect of this Purkinje cell theta stimulation on cerebellar output? For this, we recorded with electrophysiology in the interposed nucleus to see what happens during the stimulation of Purkinje cells. So this is the theta stimulation protocol as used in humans, but not in optogenetics. And then we can see here that we have this powerful inhibitory rhythmic action in the cerebellar nuclei neurons during the Purkinje cell theta stimulation. And then they restart firing, and then again we have this inhibition. So can we reproduce these therapeutic effects in rodents? Yes. And the target of the transmagnetic stimulation in patients is unknown, but we show that stimulating selectively the Purkinje cells can be sufficient. And we also reveal that preventive stimulation can exert a protective effect. Now, is the behavior effect reflected in neural activity? So for this, Berenice recorded the activity in the motor circuit, so interposed, parafascicular, so this is one of the intralaminar thalamic nucleus that projects to the striatum, and also in the motor cortex. And here you can see the results from the interpose, so this is the cerebellar nuclei output for all the groups. So one more time, levodopa does not induce any change in firing rate, so this is normal. But then you can see here that in the levodopa-induced dyskinesia, we can have this decreased activity in the interpost, and then we see a correction of cerebellar nucleus interpost firing by the cerebellar stimulations. So in the interpost, we also look at the firing irregularity, because this is something that is important and was described in many pathologies, that we can have a firing irregularity. So here I put some examples. So this is a control mouse, so you can see that they spike a lot all the time. And for example, here in the levodopa-induced dyskinesia, we can see that we have an increase in the firing irregularity of the firing rate. And then if we look, the same levodopa does not change the firing irregularity in normal animals, but then we see an increase in this firing irregularity as here. And cerebellar stimulations, they allow us to normalize this aberrant pattern of the activity that was observed in these dyskinetic animals. So then if we move to the parafascicular thalamus, so the intralateral thalamus that projects to the striatum onto the motor cortex, will have the same type of results. So here I can zoom on the motor cortex. So if you remember, so this is the moment when they are in Parkinson's disease before the levodopa, and then the activity in motor cortex is lower than in controls, as I showed in the study before. And then we see that if we give levodopa, then we'll have this increase even more than that, that is corresponding to the abnormal movements that are performed in this kinetic state. And in the parafascicular thalamus, we have a decrease. And in the two of them, we can see a correction in the parafascicular thalamus and in the motor cortex firing by the cerebellar stimulation. So overall, we see that the neural activity in the cerebellum, parafascicular nucleus, and the motor cortex is normalized after these cerebellar stimulations. Now we know that the dyskinesia is the site of the dyskinesia is in the striatum. So we wanted also to look in the striatum to see if the cellular markers of dyskinesia can also be normalized. So we turn on to look at a striatal marker that is causally linked to dyskinesia. So this is FOSB delta FOSB. This is a transcription factor with long lifetime in the cells, and it has been shown already in the field that it accumulates during dyskinesia, and even that it correlates with the score of the dyskinesia. So we look in our animals, and we could see here that we do have an increase in this FOSB expression in the lateral striatum that was impacted. And then we see here that the cerebellar stimulation normalize also this striatal expression of this marker of cellular activity that accumulates during and participates to dyskinesia. So we also show that the basal ganglia activity can be normalized. Now we wanted to go further and to understand if this effect will involve specifically the cerebelloparafascicular pathway, since this is the nucleus that goes to the striatum, and then there are other nuclei that goes more to the motor cortex, as I showed you in the introduction. And to do this, we first look into the anatomy with Jimena. So she looked at the projections from the cerebellar nuclei, projections to the thalamic parafascicular nucleus that we know is projecting to the basal ganglia. And for this, she injected retro AAV in the parafascicular, and then we could see that cerebellar nuclei, so here dentate, interpose, and fastigial, so all of them project massively to the parafascicular thalamic nucleus. So with this in mind, then we use chemogenetics to check if we really need the connection between the cerebellar nuclei to the parafascicular nucleus in order to have the effect of cerebellar stimulation on Purkinje cells. So how does it work? So these are design receptors exclusively activated by designer drugs, so with chemogenetics, and we could inhibit specifically the cells that are projecting from the cerebellar nuclei here to the parafascicular nucleus, but while we are using the theta burst stimulation. So as I showed you before, so in the preventive group, so if we start the levodopa in the same time with cerebellar stimulation of Purkinje cells, we can prevent the apparition of abnormal involuntary movement. So we have a decrease almost down to 0. And then if we inhibit specifically this connection while stimulating the Purkinje cells, we see that we do have the abnormal movements. And this means that the specific projections from the cerebellar nuclei to the parafascicular nucleus are required for the levodopa-induced dyskinesia alleviation that we obtain with the Purkinje cell theta burst stimulation. So we also saw that this is important for the therapeutic effect of Purkinje cell stimulations. So overall, in this study with Berenice, we showed that the cerebellum is an actor in the normalization of the behavior, but also of the cerebellocortical and of the cerebellar striatal pathways in Parkinson's disease. So now to end this presentation on the motor pathologies, I would like also to show you our results in the cerebellocortical pathway in dystonia. So dystonia is this neurological pathology that is characterized by the stimulation of simultaneous contractions of antagonists in muscles. So we have two muscles that can be normally are activated or inactivated, and they will be co-contracted in the same time, and this leads to involuntary movements and postures. And we know that primary dystonia can be viewed as a circuit disorder. So this involves the basal ganglia and thalamocortical and cerebellothalamocortical pathway. And we also know that there are structural abnormalities. So these are changes in the gray matter densities. We see this in the cerebellum, but also in the thalamus and the sensory motor cortex. So we also know that the cerebellothalamic alterations are present in genetic form of dystonia, and they can even be predictive of the penetrance of the disease in patients. And dystonias are associated with abnormalities in synaptic plasticity in the motor circuitry and may result from pathological learning. So we wanted to look at an animal model of basal ganglia disruption dystonia, since there are also animal model with dystonia in the cerebellum, but because we wanted to see in the same line with pathologies that develop in the basal ganglia how the cerebellum compensates and the cerebellocortical drive compensates. Then we choose an animal model of dystonia that is basal ganglia disrupted. And this is GNAL, so DIT25, which is a new familiar monogenic dystonia, GNAL. So GNAL codes for G-alpha-olf, which is important for signal transduction in the striatum. So these mice, they do not show dystonic movements when they are young, but we can induce them using muscarinic agonist, oxytremorin. So we participated to the validation of this model, and here you can see that after the oxytremorin infusion, either systemic, either intrastriatal, we see these dystonic movements and postures that are developing, and we see that this can stay over the time. So these are the heterozygotes GNAL mice. And now we ask in the team with Hind, Romain, and Laura, what are the changes in the cerebello-cerebral coupling in this animal model of dystonia? And then for this, they stimulated this time specifically the cerebellar nuclei with optogenetics, and then they recorded from the thalamus. And they did this in the presymptomatic state. So what is called presymptomatic, it's when the mice with this mutation, they do not show any dystonic movements. And then we will induce dystonic movements. And then later on, when they do not show these dystonic movements, we call this asymptomatic, since the dystonia was already induced. So even if they do not have the movements at that moment. And here we can see, so each dot is when the cells fires, and here we see the profile of the firing rate. And here in blue, you see the moment when we are stimulating the output of the cerebellum here, and the activity is in the thalamus. So we see the activity in the pre-symptomatic state. So we see already that it's increased activity. And we can see it already from this example that we have an even higher increase in the case of asymptomatic. So no movement at that moment, but dystonia already induced in these animals. And we can look at all the data, and here we can see the cerebral excitation of these thalamic neurons, and we see that we have an increased excitability. So here in the cerebellothalamic circuits in the genal mice, here, and I illustrated here to just say that this connection it's even more increased. And then in the asymptomatic post-induction of dystonia, we see that this aberrant excitability, it's even worse, it's amplified. And I put a scheme here to show that we have an even higher aberrant excitability post-induction of dystonia. So we have this abnormal potentiation in these genal plasma mice that suggests that there is a functional impairment of the cerebellothalamical pathway in dystonia, even without the dystonic movements. Now we perform also theta burst stimulation in this model. So this time we went on the cerebellar nuclei, and we performed this stimulation with the same approach. So 2 times 40 seconds per day at the theta burst. And so we had the asymptomatic state, then we performed the theta stimulation in the same time with inducing dystonia. And if we look at the data here, so here the same, each dot when the cells fire, the profile of the firing rate, and here we can see the response at the dentate stimulation pre-theta burst, and then the response after. And what you can see that, so these are the normal animals. So if it goes up, it's potentiation. If it goes down, it's depression. And we can see that we have a depression in the case of the animal model of dystonia. And we can interpret this that it's like an overexcitable pathway, as I showed you before. And then with the depression, we can have a normalization of this excitability of the cerebellothalamic circuits in this oxytremorin-induced dystonia in this animal model, genetic animal model in mice. We also look at the behavior in this case, and here we can see dystonia after optogenetic dentate theta burst stimulation. So this is the dystonia scores, and then we see here in the animal model of dystonia that we have the cerebellar stimulation have a small decrease in dystonia scores that suggest an improvement of the condition of the animal. So overall, I showed you that the basal ganglia pathologies can alter cerebellothalamic function, and also that cerebellar stimulations can correct basal ganglia dysfunctions. And now in the lab, we try to figure what and where are the plastic phenomena taking place during motor disorders. So I would like to thank all the team that we are co-directing with phenomena. So these are the ones that they did the work. To thank the founding, and thank you for your attention.
Okay, Daniela, thank you very much for this very interesting talk. And even though I know nothing about motor circuits, it was very clear, and I was even able to understand about 3/4 of it, which is great for me.
Thank you. So, I did my best.
So the floor is open to questions. Putem trece și la română dacă doriți.
Da, sigur că da.
Opresc video-ul în acest moment.
Sigur.
În continuare o să mai fie și studenți internaționali care o să participe, dar ne descurcăm.
Cine cum dorește.